if you are ever in our neck of the woods, you are welcome to come and visit. We will be happy to host you. So I'll start at the end. I'll present 3 vignettes today on the use of computational simulations to discover and characterize mutations. And so this has been work that has been ongoing in my lab since we started almost 15 years ago. And so I'll acknowledge people as we go, but a lot of the work has been done by previous group members and current group members as well. And then funding from NIH, NSF, and several private foundations. And computing time from multiple facilities is also gratefully acknowledged. We are a computational group, so we develop a lot of software. Most of it is on GitHub. Some of it we actually contribute to, so like TinkerHP, which is a molecular modeling software for classical molecular dynamics with polarizable force fields, and Psi4, which is a quantum code, the quantum mechanics molecular modeling code, are in the public domain. They're also on GitHub. We also contribute to force fields such as Amoeba and a bunch of other stuff. I'm not going to go into all of the things that we have done. I'm just going to touch on 2 of the areas of research that we cover in our group. But by and large, our main objective is to use molecular simulations to understand systems that we're interested in. So the aim of molecular simulations is to understand and predict bulk properties measured in the lab by simulating the atoms and molecules. So this means that we need to be able to calculate energies and forces of the entire system and then use statistical thermodynamics to sample these the phase space of these systems. This is a pretty famous depiction of a 3-dimensional, very rough representation of protein denaturation. So high free energy states are the denatured protein, and low energy states correspond to the native folded structure. One way to understand these systems is to use Hamiltonians, either be molecular quantum mechanics, so under Born-Oppenheimer time-independent regime, so only the electronic wave function to be solved. Another one, which is much more prevalent for simulation of the systems that we are interested in due to the size of the system, so hundreds of thousands to millions of atoms, is to use a very rough representation of the atoms and their interactions, be it bonded or non-bonded. So for example, this is the functional form of the amber force field, which the first line corresponds to the bonded interactions, so bonds, angles, and dihedrals. And then the through space interactions or non-bonded interactions corresponding to the van der Waals term and electrostatics terms. So people have been very successful using these types of classical functions coupled to a discretized form of Newton's second law to integrate over time to generate an ensemble to study different properties of a system. One of the things that we are interested in as well is also enzyme reactivity. If we want to investigate enzyme reactivity, we need to have access to electronic degrees of freedom. Like I mentioned, the systems that we would like to study, enzymes, are in the hundreds of thousands to millions of atoms. And the force fields that we use are usually non-reactive. If we wanted to study reactivity, we would need to use quantum mechanics. So how do we bridge this gap? And the answer is by using quantum mechanics and molecular mechanics, or QMMM for short. So I will present some results that have employed these techniques. The idea here is that in enzymes, we have the active site, which is a relatively small region, and this we can treat quantum mechanically, and then the rest of the system is treated classically, and then we have to have them talk to each other. This method was proposed in the 70s, 1975. The people who proposed it got awarded a Nobel Prize in 2013. It can be used for a multitude of different things to study different systems. What we have done is used classical QMMM and quantum approaches to investigate DNA damage. So DNA is subject to insults, both endogenous and exogenous. from all sorts of different things: replication stress, oxygen radicals, chemotherapy, chemotherapeutics, ionizing radiation, etc., so on and so forth. And these lead to all sorts of different damaging ends, like single-stranded breaks, double-stranded breaks, cross-links, adducts, etc., etc. So cells have evolved ways to deal with all these damage, and also replicate across damaged strands, and just replication because that's what cells need to do. So one of the things that we were wondering when we started was, what happens if during DNA damage, some of the enzymes that are tasked with fixing DNA don't do it correctly or don't do it faithfully? And so what we wanted to inquire was, are there any mutations that are associated with these DNA transaction enzymes that would be associated with a particular disease state, in this case, cancer? So we developed hypothesis-driven SNP search, or hidden SNPs. It was actually developed by the first person who graduated from our group, Rebecca Sweat, who is now a senior scientist at Relay Therapeutics. And her idea was to develop a new method that combines bioinformatics and classical and quantum mechanics simulation. So basically, what we do is we use bioinformatics to mine databases of diseases or case and control studies. We find disease-associated mutations on targeted genes. So we basically just focus on the genes that code for the proteins that we're interested in. And then for those that have mutations that are associated with a particular disease that we're interested in, we investigate structure and dynamical effects using MD, and then reactivity if needed with QMMM. So this is the main idea. And now what I will talk to you about is a couple of examples where we have applied hidden SNPs to find cancer mutations, and then another example where we have used computational simulations to develop a new anti-cancer drug. One of the families of enzymes where we have applied hidden SNPs is the iron 2 and alpha ketoglutarate family of enzymes. These are also called iron alpha Kg superfamily of enzymes. This is one particular example. This crystal structure was actually obtained at if I remember correctly, the Brookhaven light source. This is the structure of AlphaG, which is the title enzyme of the AlphaG family of enzymes, which is part of this iron alpha Kg superfamily of enzymes. AlphaG is in charge of dealkylating damaged nucleobases. So for alkylation was the first cancer treatment. So basically, if you alkylate DNA, you kill the cell, especially if you hyperalkylate DNA. Cells do have a way to fix alkylating damage via AlphaG and their human homologs. These can act on different substrates: single-stranded DNA, double-stranded DNA, RNA, even some proteins. And this is an interesting enzyme because it has an iron in the active site, but there's no heme associated with it. This iron is coordinated directly to three residues in the active site, 2 histidines and an aspartate. And this is a classical feature of all iron alpha Kg enzymes. And then this is basically an oxidation reaction. So this iron gets oxidized from the +2 state to the +4 state. And once this stage is reached, this is called a ferrule intermediate. This can be characterized spectroscopically with Mossbauer and EPR. And after this, the reaction to dealkylate DNA can take place. And this feature of iron alpha Kg enzymes is common to basically the entire superfamily. All of their mechanisms involve this ferrule intermediate, which is a stable intermediate and can be characterized with spectroscopic techniques. So we have investigated the reaction mechanism of this enzyme in detail using electronic structure methods. In the interest of time, I'm not going to dwell on that. What I'm going to show you now is how these enzymes are related to cancer. So we were interested in investigating if there are any cancer single nucleotide polymorphisms, or SNPs, that are correlated to these enzymes. Excuse me. So single nucleotide polymorphisms, or SNPs, are literally changes in one single nucleotide at the DNA level. So a C to a G, for example, or an A to a T in a region of the gene that is specifically coding for the protein. And so using hidden SNPs, we were able to search all of the genes that code for every single AlphaG family enzyme. There are 9 human AlphaG enzymes, AlphaG 1 through 8, and then FTO. FTO is a very hot cancer target right now. What we found is that there's indeed one particular cancer mutation associated with prostate cancer in one of the human homologs called AlphaG. In genomics and genetics, these single nucleotide polymorphisms are characterized by a SNP number. This RS stands for reference SNP, and a code. So RS7540 is a SNP that is found on AlphaG, one of the human homologs of AlphaG, that codes for a particular cancer mutation that changes the actual amino acid in the protein. So what we're interested in using hidden SNPs is finding a mutation in the gene, so a SNP that actually changes the final protein. So let me tell you now what that actually does, and this is where structural characterization and simulations come into play. Obviously, we need crystal structure so that we can do our simulations. So this is where light sources come in and are extremely important, and we're super grateful for our experimental colleagues. For AlphaG, what we found is that the mutation of this RH191, which gets changed to a Q in the cancer mutation, is 22 angstroms away from the active site. So the active site is located here. Arginine 191 is located over here. So what we did was we performed molecular dynamic simulations to investigate what's going on. And this is this superposition of the wild type and the cancer mutated structure after simulations. And what you can see and what we found is that the overlay is basically very similar. The root mean square deviation of the structure of the average MD of the mutant is less than 2 angstroms compared to the wild type. However, one feature that is very salient of the mutation is in the crystal structure, excuse me, in the simulations of the wild type system that has this arginine 191, that's the light gray, RH191 forms a network of hydrogen bonds that keeps this loop tightly packed together to the rest of the structure, to what's called the jelly roll fold. However, when that arginine is mutated to a glin in the prostate cancer mutant, this loop falls apart because the hydrogen bond network has been ablated. And so because of this actually doesn't have a huge structure overall, but it does have a significant impact on 2 beta sheets, these 2 here. And the problem is this beta sheet here actually holds the active site. So the active site falls apart in the cancer mutation. So we predicted, based on free energy simulations, that the co-substrate and cofactor would unbind in the mutant structures, but would remain bound in the wild type structures. And this was subsequently confirmed by our collaborators, and we published everything together some years ago now. This is difference UV-vis absorption spectroscopy. This was a technique developed by Bob Hausinger. It's an interesting spectroscopic technique because it turns out that these iron alpha Kg enzymes have a characteristic charge transfer band in the UV at 525 nanometers. When one of the co-substrates, alpha Kg, binds to the iron in the active site, we have a charge transfer between the iron and the alpha Kg, which gives rise to this 525 nanometer wavelength absorption. So what they can do is they can characterize spectroscopically both the apo structure, so no alpha Kg, and with alpha Kg. Obviously, they need to do this anaerobically, so no oxygen, because otherwise the reaction happens. So they'd have to do this in a glove box, but they can do this for the wild type, and they were also able to express the mutant. And they found that indeed, like we had predicted, there's no binding of the substrate. So what we did for this particular, excuse me, system is we not only found a mutation that's associated with prostate cancer. Using computational simulations, we predicted that the substrate which is required for enzyme activity would not bind, and we characterized that experimentally using UV-vis spectroscopy. Now I'm going to switch gears a little bit. I'm going to tell you about drug development. So another enzyme which belongs to the AlphaG iron 2 superfamily is called a lysyl hydroxylase 2. There are 3 enzymes in this family, LH1 through LH3. We're going to concentrate only on LH2. LH2 catalyzes the hydroxylation of lysine, which is found in large quantities in collagen. And collagen gives structure to cells. It's what basically forms the scaffold, so cells can have the structure. In cancer, collagen is overexpressed, and this is what gives rise to superstructures and metastasis. So what happens with LH2 is it hydroxylizes lysine, which can allow collagen to cross-link and form fibers, and these fibers extend, and thus metastasis occurs. And so one hypothesis is if we can develop inhibitors for LH2, then we can avoid metastasis. And LH2 has been also known to be overexpressed in lung cancer. So in collaboration with experimental and other theory colleagues, we developed a way to use QMMM to test inhibitors. So basically what we want to find is how these different inhibitors interact with the iron in the active site. So we basically want to plug the active site so the natural substrate alpha-ketoglutarate doesn't bind and the reaction cannot take place. So we tested around 36 different compounds. So I'm not going to go over the entire family of compounds that we tested using docking, et cetera, et cetera. All the details are in this paper. It's a series of 2 papers. But basically the idea is to have a ligand that occupies the same position as AlphaG, and the ligand can be coordinated by 1 or 2 coordination bonds. We developed an approach with QMMM to investigate the interaction between every ligand and the entire protein using polarizable QMMM. So we use a force field that includes explicit polarization. And the ligands that have the strongest interactions with the active site were the ones that we focused on and developed experimentally. There are a couple of features that we were interested in ensuring for these ligands. This is an analysis of what's called non-covalent interactions. So basically looking at the electronic density, and particularly the low density regions, we can actually use density functional theory techniques to investigate how these low electronic density regions are involved in creating non-covalent interactions like hydrogen bonds and pi-pi stacking, et cetera, et cetera. And this is done in such a way that surfaces can be developed and colored based on the type of interaction. And so, for example, here we observe that this ligand that we labeled 12 actually forms a very strong contact with arginine 728. That's this blue surface over here. And arginine 661, that's this blue surface over here. We were very interested in this R661. So 12 and 13 actually form this arginine 661 interactions. The reason why we wanted our ligands to form these arginine 661 interactions is because out of the 3 enzymes in the LH family, LH2 is the only one that has arginine at this position 661. So having a direct interaction between our ligand and this arginine hopefully would ensure selectivity, which is important because we only want to inhibit LH2. We don't want to inhibit LH1 or LH3. The other thing that we also knew from these enzymes is another feature of iron alpha-KG enzymes is because they have to be very careful with oxygen, and they're only expressed as it's what's called an adaptive response, only when damage or when they're required is detected. then they also have to be careful with oxygen. So they have tunnels to transport the oxygen into the active site so that they're not basically carrying out the reaction willy-nilly. And so what we wanted to see is if any of the, excuse me, ligands that we developed actually plugged any of these tunnels. So, or occluded any of these tunnels. And what we found is that 12 and 13 actually do. This is a difference of the tunnels. This is a reduction of 23 and 20% in the observed tunnels compared to the wild type. And for 13, a reduction of 25 and 19% in these tunnels. And so this paper explains all of these features for the tunnels. And this second paper where we actually tested these three different ligands. So this is 6, 12, and 13 that I talked about previously. These are the experimental results on the synthesized ligands using LH2. And what this shows is that the systems are selective, the ligands are selective. So for LH2, they have very good inhibition, but for several other enzymes that are very similar to LH2, the inhibition is much less. Migration assay, so because we don't want to see the lung cells migrate to other places, this is the characteristic of metastasis. If they're untreated, cells migrate with LH2 at 100 micromolar. They migrate, they don't migrate at all, and they show good killing of the cancer cells. So we're excited about these LH2 inhibitors. All three are in sub-micromolar excuse me, binding affinity IC50s. So this paper over here was just published last week. So we're excited about this one. Finally, in the last 3 or 4 minutes, I'll tell you one more story. I hope I won't take too long. We developed this database of cancer mutations of DNA repair on DNA repair enzymes. So we have a database of 8 different cancer phenotypes and 22 different DNA transaction enzymes. I'm going to tell you about this particular one. It's a lung cancer mutation on this enzyme AlphaG3H. I'm not going to spend too much time of it on AlphaG3H, except for to tell you that it's a dimer. It has a double-stranded RNA that mediates the dimerization, and AlphaG3H acts on single-stranded DNA. It's a member of the cytidine deaminase family. So every time it sees a cytosine in DNA, it will deaminate it. It will transform it into a U. It's expressed as a response against HIV. So the segment of the population that is immune against HIV is because they can express a particular version of AlphaG3H, a particular isoform that can do this reaction on HIV genomic DNA. We used hidden SNPs and we found this lung cancer variant, lysine 121E. And so Mark performed a ton of simulations. What he found is that when we have this glutamate E121 at this position, this really messes up the enzyme. So basically what happens is E121 is right here. It messes up the dimer interface. It messes up the active site. This table compares several different mutants, so single mutants and one double mutant. And what he found is that the cancer mutant is very different compared to the wild type. Every time you see a pink cell, that's a significant difference compared to the wild type reference. So he tested several different mutations in addition to the lung cancer mutation. And he also wanted to see if he could find a rescue mutation. So he predicted that the second mutation, 117E, would be a rescue. So he predicted that the lung cancer mutation would affect dimerization, would affect activity, and then this double mutation would rescue everything. We collaborated with friends in Canada, in the lab of Linda Chelico at the University of Saskatchewan in Saskatoon. And all of the predictions that Mark did based on his calculations. So expression, the 121E is not expressed, the double mutant is expressed at the same levels as wild type. The control 117E is actually overexpressed. They were very excited. Activity, the double mutant is active, is 4-fold less active than the other ones, but it's still active, and it dimerizes. So everything that he proposed was correct. They also did an assay to investigate if the mutation was actually involved in lung cancer proliferation. So these are healthy cells. These are lung cancer cells. And lo and behold, the 121E mutation is indeed involved in lung cancer proliferation. So with that, I'm going to give you the summary. So we have this new approach, hidden SNPs, that combines bioinformatics and atomistic simulations. We found a prostate cancer associated mutation on AlphaG that affects substrate binding, developed a promising anti-cancer LH2 inhibitor for lung cancer, and we found another mutation on A3H, which is associated with lung cancer. With that, I'll thank everyone again. And I guess we have 4 minutes, so that wasn't too terrible. And I'll be happy to answer questions. Thank you very much.
Thank you. Very nice talk. Are there any questions for the speaker? Wolf Dieter.
Hi there. Interesting talk. Just for the last one, I didn't quite get the motivation why a single mutation. In both cases, it's a positively charged residue replaced by a negative. And if you've only got the mutation in the 121 position, then you say it's inactive and the protein can't be expressed. Yeah. But when you do a double mutation, then it works again. I didn't get that.
Yeah, sorry, I flew through this one. So, the feature of this particular position 121 is that there's actually 2 positive residues, 2 lysines, right? So the native structure has K117, K121. So those are repulsive interactions. So when you only have one at 121, one change there, that creates a whole new network of hydrogen bonds. So that's what is here. So all of these numbers are increased. That's an increase in the hydrogen bond. So that tightens everything. So it tightens all of the structure close to that goes to the dimer interface, and it tightens the structure going into the active site. And we actually characterize that with network analysis and normal mode analysis. And so you can see it here. This is the first normal mode comparison. So the gray in the back is the wild type, and the blue is the particular mutant. So in 121E, you see that the comparison between mode 1 and mode 2 is much tighter for the cancer mutant than for the wild type. But for the double mutant, the dynamics are much more similar, and 117E is much more similar. So it's because of that. So the hypothesis for changing this to a glutamate was to restore the repulsion between sites 117 and 121, which would remove those new hydrogen bonds.
But you would still have the salt bridge. I mean, essentially, you're incorporating new salt bridges. But your hypothesis is that it creates new salt bridges and that lock the protein into place, and then it loses its dynamic.
It abolishes actually one particular one really important salt bridge is this one between the backbone of 117 and 121. So when you make a repulsive electrostatic interaction between 117 and 121, this is broken.
That's not a salt bridge, though, is it?
It's a hydrogen bond. Sorry, between the backbone. That is correct. Yeah, but that's what happens. This particular 117 to 121, which is here, is returned to wild type. Goes from 65 to 56%. Okay.
So yeah, it's quite complicated, but
it is, yeah.
And but then why wouldn't it be expressed at all? That seems really strange, because by including a new salt bridge, it should actually stabilize the protein, and then you should be able to make it easily. And then,
but the problem is the monomer is created, but now it cannot dimerize, right? And so for the expression, I don't remember if
but the monomer should still be produced.
The full dimer or just the monomer in this expression.
It might be if it's the dimer.
Maybe it's expressed, but just it's beyond the detection limit. So,
okay. Good, thank you.
Okay, let's reward the speaker with a hand clap.
Thank you very much.
Hands up. And then what we can do is come at the end of the session, come back to field any more questions if the speaker is still around. Or you can put a question in the chat, and if he's not around, we'll try to email them. And you can get the speakers' email addresses from the conference webpage. Okay, so moving on to the next speaker is Lucas Paul. Lucas, I did see your name in the gallery earlier. Lucas, are you here?
Yes, I'm here. I'm here.
Okay, please share your screen and begin.
Okay. Just a couple minutes. Okay.
Go ahead. Yeah.
Hello, everybody. I think you should be able to see my screen and
yes, and go to presentation mode, and then you can start. Yep, there you go.
Yeah. So thank you very much for this opportunity to present to this conference. I'm honored and I'm humbled for this opportunity. Yeah. So my name is Lucas Paul Chemba. I'm from University of Dar es Salaam, here in Tanzania. I belong to the chemistry department, and I graduated in my PhD just recently, last year. And I'm here. I have established I have the computational group I'm working with the 4 master students, and I have colleagues, of course, whom actually we are collaborating to enhance this computational chemistry. So I'm going to present to you our study actually, which was a bit kind of collaboration, and it was a result of training of the master student with my colleague, Professor Andrew Poruch from Miami University, who actually I worked with when I was working with my PhD. And he assisted me with the solvation free energy technique, which actually was new to me. So after our interaction, I was interested to see if he can support us to give the basics and how to work with the solvation free energy. And of course, we are humbled because he provided with the opportunity to access some facilities in his university in the US. So this was a work after like a 6-month training, and we ended deciding of developing a project which actually could be like our end product of our training. So we wanted we are interested to investigate the structure and the stability, also the solubility of psilocybin in the water, and the pure organic solvents. This is a molecular simulation study. So this is my presentation outline. I would like to introduce this psilocybin by highlighting some of the terminology like the psychedelic, which actually is a subclass of hallucinogenic drugs whose primary effect is to trigger the non-ordinary mental states, which actually is known as psychedelic experience or the trips, and an apparent expansion of the consciousness. But also the psychedelics can cause the person to hallucinate, seeing or hearing things that do not exist, or things which are distorted. So the hallucinogenic are psychedelic drugs that are potentially what can potentially change the way people they see, they hear, they taste, they smell, or sometimes how they feel, but also affect the mood and the thoughts. So psilocybin, which is our compound, is actually is kind of magic natural occurring, and they are consumed for their actually for the genic effect. As I have said, this compound, they are from the mushroom. And when we are interested to see or to investigate the psilocybin, most of the literature they present, they express the psilocybin. It exists in two different forms. So this is the two forms of psilocybin which I'm going to present to you. We have psilocybin A and psilocybin B. So we decided to label psilocybin A and B. So these are two forms of psilocybin. But if you see, when it is consumed, this this psilocybin, it undergo the dephosphorylation and it end up with psilocybin. This is actually which is like an end product. So by from this from the expression of the group, you can see there is substitution of those. Of course, it goes. It explains itself like the phosphorus group with the other atoms. They are eliminated, and it goes with the hydrogen. Hello. Oh, okay. My cursor was okay. My slide was not moving. Okay, so as I have said, psilocybin is an indole-based from mushroom. It was first isolated in 1957, and actually it was synthesized in 1958. It has a wide range of pharmacological application, like the neuroprotective effect, the mental disorder. It reduces anxiety, and it has the hallucinogenic activity. For more than 1 decade, psilocybin has been approved by FDA for clinical application. So, and more studies demonstrate that psilocybin has a great impact on the treatment of the psychiatric disorder. And recently there are a quite number of literature which they have been reporting this psilocybin. Like this is the last year document which was reported about the single dose psilocybin for treating resistance episode major depression, and another literature which was looking at the effect of psilocybin assisted therapy on major depression, depressive disorder. More literature they have been reporting. They are still reporting about psilocybin, and even the experimental experiment they are still going on about the application of this psilocybin. So, despite of being the potential drug for different application, it suffers from sort kind of limitations to its application. One of the major challenge is the poor solvent solubility, but also the neurotoxic activity of psilocybin. But for quite a number of years, psilocybin again has been legally restricted to be applied in the medical field, but also the scientific research. So to address the challenge, psilocybin need to be dissolved, or we need to find the appropriate solvent, which actually psilocybin can be extracted, can be we can actually work with psilocybin because of poor solubility. But also the application of other carriers like nanoparticles can be maybe applied to work with psilocybin. So for this study, we intended to investigate the solvation-free energy of psilocybin. Actually, we went to see with the commonly used the industrial pharmaceutical solvents, and we used the 35 commonly plus water. And why do we need the motivation, why do we need the solvation-free energy? Why should we talk about the solvation-free energy? Yeah, it's important because it helps to understand the drug solubility in different fluid, but also in the different solvents. But also it actually informs us about the bioavailability, actually to see the ability of the drug to dissolve the body. But also it helps to determine the dosing regime of the drug. Moreover, it helps to look at the formulation of the drug into various dosage form, like tablet, capsule, and maybe it can be used to by injection. But also it determines the distribution and elimination of the drug in the body, like to what extent the drug is going to stay in the body, or to what extent the drug is going to be eliminated. But also it goes to the read optimization after we have identified the solvent. We have identified the compound. It goes to the read optimization. But also the safety and the toxicity is very, very important. Moreover, it helps the regulatory authority to consider. It has to do with, they have to check what actually it's one of the criteria for most of the regulatory authority to consider. So our motivation, as I have said, was to find which form is more preferable, because like when we want to work, like anybody wants to work with psilocybin, actually will face the challenge of these two forms of psilocybin, which they do exist, actually which are the totem of each other. So it was very important for us to see which one actually is maybe is thermodynamically stable, or it's most favorable, which actually psilocybin, as it exists in two forms, which one is more preferable form of the two. So we, as I have said, we used the solvation-free energy. This approach, it look at the drug in the ideal gas, and the transfer, the Gibbs-free energy of transfer from the ideal gas to the solvent. And again, if I have to explain about this this figure, we have solvent 1 and we have solvent 2. So the Gibbs-free energy of transferring of the drug from the ideal gas to solvent, like solvent 1, you're going to see what the is the drug, does it prefer to interact with the solvent, or does not prefer to interact with the solvent molecules. But also, if we have solvent 1 and we have solvent 2, then we're going to see how the drug will prefer to go to solvent 1 or to go to solvent 2. Maybe if it has to move from solvent 1 to solvent 2, then the Gibbs-free energy of transferring of the drug from solvent 1 to 2 is now determined. Then for our psilocybin from A and B, then again the same diagram can be used here. As we have the ideal gas of the two forms from A and B, then we have to see the conversion of psilocybin from A to B, or B to A in the ideal gas, but also the transfer or the interaction of from A in the solvent, but also the conversion of from A to B in the solvent. We use the methodology, as I have said, the solvation-free energy. And here, the class 1 potential energy function was used to model all the interaction. The Jones potential was considered for non-bonded interaction. And for our calculation or for our simulation, actually, we use the single psilocybin, whether from A or from B, which was also was dissolved in water and the other certified organic solvents. And the force field which was used was the GAF as implemented in the amber, and the parameter generated was generated by antechamber, then it was converted from amber to Gromax, and the water model which was used was TIP4P 2005 model. And the packing of the initial structure, we use the pack model to generate the initial structure, and we learn the 300 steps to remove any bad contact from the packing. Then there are 2 steps of equilibration was performed at NPT ensemble by integrating the equation of motion. And the first was the 2 nanosecond equilibration, and the second was followed actually by 12 nanosecond equilibration. And the final structure of this series of simulation were used as the initial for the solvation free energy calculation. And the free energy calculation, we use the multi-state Bennett acceptance ratio method to calculate the solvation free energy. And the soft core potential was used to decouple the solvent, the solute or the drug, and the selected solvent molecules to look at the intermolecular interaction. And the stage of dependent decoupling parameter was controlled by the LJ and the electrostatic intermolecular interaction. And actually, the decoupling parameter was varying from 0 to 1. The 0, which implies there is no any kind of interaction, and the 1, which implies there is full interaction. And here are the results for the solvation free energy. From the findings, it can be observed that we calculated the total energy, the LJ interaction, and the Coulombic interaction, and comparing the 2 forms of form A and form B. You can see that in all cases, contribution of for the total energy for the form B, they are less than form A. This indicates that the greater stability of form B in solution compared to form A. And to look more for the analysis, we performed the structural analysis. Because psilocybin, as I have said, it has some different groups, some different atoms. Most of the atoms, they are polar. They are polar atoms, which we expect that whether from psilocybin from A or from B, those polar atoms will be interacting with the polar hydrogen from the solvents. So we were interested to examine their structural analysis. And actually, we performed the structural analysis. One of the parameters we wanted to see, it was the connection matrix analysis, which is a CMAT analysis, and we use the Travis software to do this task. And this one, this structure actually represent among, I just picked one from the analysis, but I use this as a template to explain, then I'll go to the result of the CMAT analysis. So we have the left part of our diagram, which it has the electron, the polar atoms from, they can be they can be whether from the drug or from the solvent. So the column we have N1, N2, oxygen 1, oxygen 2, 3, and this oxygen 4 and oxygen 1. So this labeling will be specifically for the psilocybin in different solvents. Again, this part it shows there is arrangement of the polar atoms, but also we have the hydrogen. They can be also from the drug like psilocybin, or they can be hydrogens from the solvent. And the right-hand side is the heat map, which give the key information like the red color implies there's a strong interaction between the specific polar atom and another hydrogen, and the blue it implies that there is short distance, but there is poor interaction, and the yellow color implies that there is high interaction, but at a long distance, because this heat map it shows the first maximum height and the first maximum distance. So it combines the two. So the first analysis, we did most of the analysis, but I'm going to present just a few solvent which for this presentation. So for the CMET analysis, we did also for acetone. As you can see, acetone has a polar oxygen, and there's two forms of psilocybin. We expect like the oxygen from A and from B, but also we have nitrogen, but also we have the nitrogen, hydrogen from nitrogen in two forms from A and B. This actually is the findings in acetone. So we have oxygen from acetone, oxygen 1, and we have actually we have the polar from psilocybin here, and we have the oxygen from acetone, but also we have the hydrogens from psilocybin. We just labeled them, and of course this numbering was actually generated from the Travis software. So when it can be compared to form B, when it can be compared to form B, can see for form A, we have strong interaction between two hydrogen 1 and hydrogen 2 in psilocybin form A, which they were strongly interacting with oxygen 1 of acetone. But here with form B, there is only strong interaction. Actually, there is even just poor interaction between hydrogen 1 and hydrogen 2 of psilocybin, were poorly interacting with oxygen 1 in acetone. And actually, this hydrogen 2 of psilocybin, there was actually intramolecular interaction between the hydrogen 2 of psilocybin and the oxygen 3 and the oxygen 4 of the psilocybin itself. And the next analysis, I also considered the ethanol in both forms, from A and from B. It can be seen clearly that ethanol has the polar oxygen, but also ethanol has to donate a proton, which is actually labeled hydrogen 1, which actually the two hydrogen 1 and 2 of psilocybin had somehow strong interaction with oxygen, 1 of ethanol, but this hydrogen of ethanol had a strong interaction with oxygen, 4 of psilocybin. But this that behavior also was consistent when you check for the interaction with the psilocybin from B. Also hydrogen from ethanol was strongly interacting with the oxygen 3 and the oxygen 4 of psilocybin. So this actually was a structural analysis to see how the 2 forms of psilocybin they are behaving in different solvents. So another analysis was the radio distribution function from A and from B. Sorry, yeah, here it has to be from it has to be from A and from. So we also presented the radio distribution function in some of the selected solvents, but also from these findings can be seen that the radio distribution function in form B, they are quite large compared to radio distribution function from A. Like we use the same like acetone, but because from the CMET analysis, it was very important. We observed some of the interacting atoms, like a certain hydrogen was interacting with a certain polar group from psilocybin or from solvent. So those strong interacting atom, we were interested to see how are they behaving in lab? But generally, you can be most of the RNA in form B.
You should probably get to the conclusion. You have like about 5 minutes. There you go. Thank you.
Okay. Yeah. So I'm about to conclude. So just to conclude, psilocybin is an active psychedelic compound which actually is known for hallucinogenic property, which actually is present in the magic mushroom. And since its discovery in 1959, psilocybin has been studied in clinical trial and it holds a significant promise for treating mental health disorder. And but also the legal restriction has hindered its progressive research in medical field. So the results from our findings, we concluded that psilocybin from B, which actually we labeled it from B, is thermodynamically favored tautomer among the two, or compared to from A. And from B has higher stabilization in solvent than from A, but also from B has strong hydrogen bonding by accepting the proton than donating, as we have observed, and it has more strong interacting with most of the alcohol and the water. There is an opportunity for anti-solvent crystallization to do the purification, and anybody who is interested to work with psilocybin. So working with psilocybin for our computational study, we wanted to see, even if it is legally restricted, but when maybe in the future, this restriction, when it's going to be removed, then people, they have to have some basis of doing the research working with the solvent for working with the psilocybin. I think this marked the end of my presentation. I did appreciate my collaborator and other people whom I'm working with. And of course, this work we performed in Ohio Supercomputing Center, and some time I worked with when I was in PhD, I worked with South Africa HPC, which is called Lengau. One of our colleagues has access to this center. So thank you very much for your attention, and this is my work. Thank you very much.
All right, thank you for a very nice talk. Are there any questions for the speaker? Chart, I see you. I see your hand. We have time for one, maybe two questions. Chart, was your hand up? Oh, that was a clap. I'm sorry. Trevor, did you have a question?
I'm fine.
Okay. All right. Well, why don't we move on? The third speaker is here now. But let's thank the speaker real quick right now. There's a nice round of applause and hearts and flowers. And then our next speaker here is Dr. Philip Kurian from Howard University in the United States. Philip, let me make you the host. I think you can go ahead and share your screen. Yep.
Okay. Thank you so much, Lawrence. Okay. Thank you, Lawrence. Thank you, Chart, and all the organizers of this wonderful conference. As Lawrence mentioned, my name is Philip Kurian, and I'm principal investigator and founding director of the Quantum Biology Lab at Howard University in the U.S. Capitol. So today I'm going to give you a high-level overview and focus about quantum biology and its implications for understanding the computational capacity of living systems, and even pointing to the computational capacity of all matter in the observable universe. But I'm going to do that by focusing on a specific project that is now published to the archive, but in review. So recent work this year, and done by two of my postdocs, which I'll highlight in a moment. So for those of you who aren't familiar with quantum biology, I want to clarify that this term as an overarching field has many different meanings to many different people, some much more technical and experimental than others, and some more broad and thinking about what are the implications of the quantum theory to living systems. And there's a reason for that. So, okay. So in theoretical physics, that's my training and background. We like simple models. And in particular, in the realm of quantum optics, meaning the interaction of light and matter, we particularly like solvable models like two-level systems. And as you can see here, we have a two-level system where you have an absorption and an emission, which I'm sure many of you are familiar with. And we like that because it's one of the few models in physics that is analytically solvable. But however, like the harmonic oscillator and other models. But however, as we know, reality is not the same as physics, and physics is not the same as reality. And this is consistent with John Archibald Wheeler's view in this wonderful little essay that he wrote 40 years ago called Law Without Law, where he called physics an elaborate construction of imagination and theory fit between a few posts of clear observation. And he even called it a paper mache construction because, you know, as you can see here from this picture from his essay, we do have these few posts of clear observation, but there are many ways to connect these posts, right? And in the absence of evidence, right, there are some more inventive and elegant ways of connecting the posts than others. So now, just a few months ago, actually, I discovered there's not only spherical cows, which physicists are fond of, but also spherical birds. And so this is actually a bearded reedling, and the avian method of navigation has gained a lot of press for its relationship to quantum entanglement, spin entanglement between electrons and nuclei in a molecule called cryptochrome, and others. But the challenge for understanding how delicate quantum effects like entanglement could be implicated in something as complex as a living organism poses many challenges. The colloquial way of saying it is it's messy. Living systems are messy. They operate in nonlinear, non-equilibrium, and systems that operate far beyond the Born-Oppenheimer approximation. They are non-adiabatic. And this poses some challenges for treating things like in a Brownian motion or statistical thermal equilibrium environment. So the other question is all these different orders of magnitude that biology seems to synchronize, even at the quantum level, all the way to the organismal scale. So we have to deal at some level with how the quantum vacuum and these quantum transitions occurring at the molecular level scale up to actually deal with how a collective system organizes and communicates and synchronizes. And so while we don't have that previous picture of the quantum vacuum, this is the paradigmatic model of light and life, starting from the atom and then moving out to the cosmos. So in 2017, my group published this work on oxidative species-induced excitonic transport in these aromatic networks that we see in many living systems, higher organisms. So the picture to have in mind is it doesn't have to be a neuron, but neurons have a host of these mitochondrial networks. This is actually a cartoon, but of course in neurons and in other filamentous structures, you actually have reticula that are made from mitochondria. And these mitochondria emit these molecules called reactive oxygen species, which are produced by all cells in the body and in living organisms that are undergoing oxidative metabolism, which is what you do when you breathe in oxygen and respire carbon dioxide. So turns out these reactive oxygen species emit specific wavelengths of light called ultra-weak photon emissions. And as it turns out again, these wavelengths of light are actually well-tuned to many of the chromophores that occur in biology. But for generations, even maybe centuries, people were considering that these ultra-weak photon emissions, or biophotons as they were called, were just junk, kind of thermal heating up the cell. But recent work of ours has suggested a different picture. So the physical picture you want to have in mind is that of if you have a single molecule that has a two-level system or a single excitation or excited state, imagine putting them together and coupling them through some interaction, Coulombic or otherwise. And you have these aggregate states. You can call it upper polaritonic, lower polaritonic. You can call it superradiant, subradiant. There are many different names. You can call it high and low, highest occupied, lowest unoccupied, right? But these molecular resonances that are on site, meaning at the excitation wavelength, can be red shifted and blue shifted in the aggregate states, okay? So now imagine you don't just have two, but you have several and large architectures of these networks. And in fact, you can write a coherent superposition of these where the first molecule is in the excited state, all others in the ground state, and so on, where the second is in the excited state, all others in the ground state, all the way to the end molecule being in the excited state, and so on. And this is what's called the single excitation manifold. And that's also exactly solvable under certain assumptions. And so this has actually been well documented in high-profile publications by Martin Scully's group, as well as Asenjo Garcia's group more recently in Nature Communications. Now, This system of two-level systems is actually an open system, and these open quantum systems are widely used in the quantum computing literature to discuss systems of two-level emitters. But when you deal with an open system, of course, probability is not conserved, so you have a decay out to the electromagnetic field. So we have a non-Hermitian Hamiltonian, so you have to be careful. There's some differences with Hermitian quantum mechanics. And so these non-Hermitian systems, you can see by these complex values here, these actually admit couplings when you derive. So I'm not showing the derivation, of course, but these couplings, omega and upsilon, arise from the collective interaction of the system of two-level systems with the electromagnetic field. So you'll notice, in addition to the standard transition dipole coupling that goes as 1 over r cubed, you have other terms, 1 over r squared, which admit long-range interactions. And so these can also be rederived in a retarded or non-retarded framework. And so you can see where these terms emerge. And so some of you may be familiar with Forster resonance. So Forster resonance is a short-range coupling that you get when you take the 1 over r cubed, you get 1 over r cubed from the other, and you get 1 over r to the 6th. But these are much stronger couplings at long range. Now, with non-Hermitian physics, you get complex valued eigenspectra. So you have an energy eigenspectrum and a number of decay rates. And these you can think of as the decay rate of the collective. And Robert Dickey in 1954 showed that in systems of these two-level systems, in a gas, for example, you can see these superradiant states where big gamma J is much, much larger than little gamma. And we will see this emerge now, not only in a gas, but in systems of interest to biology. So in 2019, we published this theoretical work. It was purely theoretical, doing this modeling of the transition dipole network in microtubules. And microtubules are made of the protein tubulin, shown here about 5 nanometers across, and then shown here in its polymer at 25 nanometers across. And these structures form organized networks that have this spiral cylindrical symmetry. You can see it's a cylinder with an offset. Now, we were surprised to find that if you only look at one spiral, I know it's hard to see here on the slide, but if you only look at one spiral, you don't see much superradiance. You see 15 times the single tryptophan decay rate, and that's modest. But at 10 spirals, as you repeat this structure, you have more than 150 times the single excitation, single tryptophan superradiance. And then at 100 spirals, you have 600 times that single trip superradiance. And if you were wondering if this is just the result of any repetitive sequence, we took 100 spirals, put them in that same order, but randomized the orientations. And you can see that at 100 spirals, this washes out the superradiance. So this was an intriguing experimental find that also manifests this geometry in photosynthetic complexes, but in the visible spectrum. So there's many similarities between the arrangement of these complexes that absorb in the ultraviolet and those in photosynthetic complexes or nanotubes, photosynthetic nanotubes that absorb in the visible. Now, if you want a picture of how these look, we plotted the wave function of finding the exciton, or so the probability amplitude squared of finding the exciton on a single tryptophan of this microtubule segment with 10,400 chromophores. This is for a superradiant state in lateral view and in cross section. So here's the more recent work that combines experimental work with our collaborators, Majed Chergoui's group in EPFL in Switzerland, and Luca Cilardo's theory group in Florence, and our colleagues in Mexico who are part of the QBL. Okay, so these networks of tryptophan that I described in microtubules and in tubulin, they exist actually throughout biology, and they form these large hierarchical networks in transmembrane proteins, photoreceptor complexes. There's actually one called UVR8, which specifically uses its arrangement of tryptophans to funnel energy in plants. They're found in subneuronal elements like axons. They're even found in viruses and in other cellular organelles and appendages. So if you want to take a closer look, here is the tubulin dimer, which has 4 tryptophans in each monomer, total of 8. You have a microtubule, you can see here, built from the tubulin. It's got 104 tryptophans in a single spiral. And then you've got this structure like the axoneme, which shows up as the structural core of cilia and flagella, the centriole, which organizes cells in a 2 to 3 degree range in its azimuthal plane, and then axons like this, which form huge bundles, half a micron or more. These are rather tiny ones shown here. So here are our predictions. These were predictions that we made at thermal equilibrium. And you can write this using the Boltzmann distribution, where the probability that the trip network is described by an energy eigenstate indexed by K at time t. And this is in thermal equilibrium. So we're choosing an environment that is extremely hostile to the maintenance of quantum effects. Now you can take the thermal average of the quantum yield, which you can describe as the number of photons emitted over the number of those absorbed, or you can rewrite it in terms of the radiative and the non-radiative decay rates. Turns out that for a non-Hermitian open quantum system, you need to describe the decay rate as leaving the system. So the probability is not constant, but you can then write the thermal radiative decay rate as a sum over our radiant states weighted by this Boltzmann factor. We were surprised to find that starting from the reference standard of tryptophan in solution, we actually get an increase in all these structures. Of course, in the microtubules at 10,000 tryptophan, that's 100 spirals, we begin to see saturation. But in the centriole and in a 91 microtubule bundle, we do not reach the saturation regime even at 100,000 tryptophan chromophores. So this is experiment. So all the slides that have experiment are going to be EXP in the upper left corner. So these are our steady state measurements. So nothing fancy experimentally, but this is the steady state measurements of quantum yield that do agree with our predictions at thermal equilibrium. So here's the result. Here's the steady state absorption spectrum for tryptophan, tubulin, and microtubules. And here are the Stokes shifted fluorescence spectra of those same compounds. Now, I want to highlight for you that there are similar absorption maxima for all 3, but the absorption emission Stokes shift is much different between tryptophan in orange and the microtubule and tubulin. So it's much smaller for these 2 because of the protective protein environment around the tryptophan chromophores. Now, we determined the quantum yields at both 280 nanometers, where many chromophores absorb, and at 295 nanometers, where only tryptophan, the tryptophan is the only amino acid that absorbs at 295. So we've subtracted the scattering background of the microtubules according to a Raleigh model, and the mean tubulin spectrum here is plotted in red for comparison, and you can see excellent agreement there. So we've been able to then, I want to highlight your attention to these quantum yields that we calculated. So from tryptophan to tubulin, you see a decrease in the quantum yield. It's not necessarily statistically significant, but at least for our 5 samples of each done in multiple trials, we were able to show that it decreased. This indicates that there is, as you go from water to the protein environment, there is a increase in the non-radiative decay rate, the channels that are not being emitted to the electromagnetic field. However, when you stay in the protein, but then you increase in microtubules, notice that the quantum yield increases between 50 and 90%, or 50 and 70%, depending on the wavelength of excitation. And this is interesting because without the non-radiative decay rate decreasing in the protein, you then have this enormous increase in the radiative decay rate that swamps the other contributions. So what is causing this increase? We argue that this is due to the superradiant effect of the collective state. So it turns out that longer centrioles exhibit this cooperative robustness to static disorder. So if you're wondering, how does the superradiant effect change? You see here, we have almost 4,000, more than 4,000 times the superradiant decay rate when you put 27 microtubules in the conformation that you see here, which is found in biology in centrioles. And you see that again here displayed in the spectrum in the absence of disorder. As we add disorder, though, you can see that as you go from smaller centrioles, shorter centrioles at 64 nanometers, you see a sharp decrease as you increase the disorder in the system. However, contrary to conventional assumptions, as you increase the length of the centriole, you make your system bigger and bigger. The cooperative robustness of all these states together enhances the quantum effect. And you essentially get a more robust state that is more robust even at higher disorders, okay? Remembering that 200 inverse centimeters corresponds to KBT at about 288 Kelvin. And you see here that even though it's not at 200 inverse centimeters, it's not an enormous enhancement. This is an order of magnitude enhancement over standard optical effects in the biological milieu. So if biology has access to this physical effect, it stands to reason that it might be using it in evolutionary processes. Now, why is the quantum yield so important? So we looked at the robustness of quantum yield to disorder. And as you can see here, we reproduce this for extremely large values of the static disorder in microtubules and centrioles. And the key point to recognize is that even at 500 inverse centimeters, this is far beyond thermal disorder in a biological environment, you still see the cooperative enhancement, even a little bit at 1,000 inverse centimeters. And so this is remarkable in such large structures that you would see this enhancement even at high temperature. So this arises from the presence of superradiant states that are close to the lowest excited state, remembering that in this Boltzmann distribution, those that are close to the ground state or the lowest excited state are weighted more strongly than those at high energy. So of course, the fact that we have biology is selecting for these states that are close to the lowest excited state, this enhances the quantum yield. Now, why might that be relevant? Well, if biology can absorb photons at a higher energy, down convert the energy, and then emit via Stokes shift, then this would provide a way of taking highly energetic UV photons and making them less damaging to the biological milieu. So here were time-resolved experiments that we did. And what you should notice is that this is on the picosecond timescale. So on the picosecond timescale, notice the kind of flat decay of tryptophan out to 3 nanoseconds, which we don't show here. But notice the ultra-fast decay of tubulin and microtubules on the 5 to 10 picosecond timescale, which is what we predicted in the event of superradiant states in these structures. So this remains to be seen, but we need to compare the extremely short time component, the sharp initial decay, with this long subradiant time component that we think may be even longer than the tryptophan decay. So without going into too much detail because of time, we see superradiance in the UV as you build these microtubule bundles that are realistic and found in mammalian neurons. And so you can see here, you see several thousand times the superradiant line width of the single tryptophan, only when you're at 91 nanometers of axon length. So this is remarkable that as you pack them into a three-dimensional space, you get more advantages within, let's say, the excitation wavelength of tryptophan, which is about a peak of 280 to 300 nanometers. And so this extends even further as you go to longer or larger structures built with larger numbers of microtubules. So in principle, you can get, in the absence of disorder, up to even 10,000 times the single tryptophan decay rate. And we have a analytical form from these numerical simulations that we've diagonalized with several 100,000 by 100,000 matrix elements. No, we also ended up building a stoichiometrically accurate model of SARS-CoV-2 and the exterior tryptophan network on SARS-CoV-2, which is very similar to some of the other coronaviruses, but has an unusually hard shell and ordered structure. So we built this. It took quite a long time to build this from PDB files and to build it using 4 different structures and then to make it stoichiometrically accurate. So this is a full model of the exterior tryptophan network. And we were surprised to find as well, and 1,000 times at least, the superradiant effect due to this collective state around the system. So we're currently pursuing this as a detection and discrimination method with coherent Raman spectroscopy in the vibrational degrees of freedom to look at SARS-CoV-2 compared to other viruses. And you can see here, compared to enterovirus and rhinovirus, there's an enormous superradiant effect and a superradiant redshift relative to these other more common viruses. Now, how much time do I have, Lawrence? Or anyone?
5 minutes.
5 minutes, great. So in the last few minutes, I just want to summarize kind of what some of the effects, these quantum optical effects, may have on revisiting our conception of information processing in biological systems. So I refer you to the archive preprint that is based on this work, but this table that is in the archive preprint highlights that there are 2 timescales in these collective quantum optical systems that I've described here. The superradiant timescale on the picosecond scale, and then the subradiant timescale on second and longer timescale. So this is interesting because these collective quantum systems have inbuilt these divergent timescales. So the idea that quantum effects only occur or can only be observed on very short timescales is in some sense a limiting way to view it. So this is a conventional calculation for the lower bound computational speed for the human brain. We have about 10 billion neurons operating at the kilohertz timescale, which is the chemical timescale for Hodgkin-Huxley signaling in neurons. So that gives you 10 to the 14th ops per second. Now, if you take those 10 to the 11th neurons and you say, oh, well, if I have these quantum optical effects operating at the picosecond timescale, then potentially I have terahertz information processing. So this exceeds exascale, 10 to the 23 ops per second. Now, the energy consumption requirements of the brain do not scale exactly like classical digital computers. Why? Well, the Frontier supercomputer, that's our state-of-the-art supercomputer at Oak Ridge, they operate at exascale, which operates at about 20 megawatts. Now, in the human brain, we have beyond exascale, almost 6 orders of magnitude beyond exascale, granted for short times, but we operate on less power than a 60-watt light bulb. And this is using many more kilometers of axons and dendrites if you were to extend them out. So keep this in mind, noting that you have on the time scale of these superradiant states a power output on the order of microwatts. So multiplying that by 10 billion, you can see how this scaling of the brain to the supercomputers matches, okay? And here, notice that on the subradiant timescale, even for some axons, we have timescales of tens of seconds and even longer. So you have quantum effects in the long time regime that are observable even at long timescales. So I won't go through this in detail, but I refer you to Seth Lloyd and Paul Davies' work. But from merely the Heisenberg uncertainty principle, we can derive the maximum number of operations per second of observable matter in the universe. So for those trainees out there, I encourage you to do dimensional checks, and those dimensional checks provide a nice reference point that you're doing your work correctly. So we can take estimates for the volume of the observable universe using the universal speed limit set by the speed of light. And we can then derive the maximum ops per second of the matter-dominated universe. Now, so here's a nice formula for the matter-dominated universe. Now, we can also take this even further. Using some of the estimates that I've made, we can also determine the computational limits of all life and all biomass on Earth. So we use data from the WMAP satellite and the Hubble telescope. We can use simple basic Newtonian physics to then get us to a derivation of the critical matter density of the universe. And then noticing that we use that formula we derived on the last slide to get this beautiful formula that comes out with the Planck time. So it stands to reason that if you divide the time of the universe into Planck time segments and then square it, right, due to the spherical nature of the universe expanding, you would get something that corresponds to the information processing capacity of the universe. So it turns out we have 10 to the 122 total operations in the history of the universe. And I'll leave this for your work. All kingdoms of life on Earth, assuming quantum optical information processing as a maximum, can have performed no more than about half of the total operation, the square root of the total operations in the history of the universe. So the idea that quantum effects play a role in living systems changes our understanding of life's information processing capacity in the context of the cosmos and the observable universe. So just to wrap up, here's what the Quantum Biology Lab does. We're at the nexus of theoretical physics, applied maths, and biological and chemical physics. And here are some of our recent projects. We work in slime mold, doing self-organization, sensing, and agency, but we also do molecular dynamics simulations, quantum optics effects. We study non-Markovian processes. We also discuss many body dispersion effects, Casimir effects emerging from the quantum vacuum in DNA enzyme systems. And we have a K-12 liaison that helps us to develop virtual reality curricula to teach K-12 learners quantum concepts. So I leave you with this quote from Max Planck, who encouraged us to explore not only the physical world, but worlds beyond it. And so if you're interested in joining the QBL, please reach out to us on our website, which you can find at quantumbiolab.com. So thank you so much for your time, and I look forward to your questions.
Thank you for a very, very interesting talk. Are there any questions for the speaker?
Thank you, Lawrence.
Sorry.
Yes, thank you, Lawrence. And thank you, Philip, for a very interesting talk. It's really fascinating. Yeah, everything that you're involved with and also the work that you presented. So I have a couple of questions on the modeling that you performed on the tryptophan in the microtubules and the centrioles. So obviously the spectroscopic parameters of any molecule, tryptophan in this case, depends strongly on its microenvironment.
Yes.
So did you treat all the tryptophans the same? And if yes, how realistic is this assumption?
Yes, so we've done it in many different microenvironments. So the main parameters of the model are the 1LA excitation of tryptophan. So that is the emitting state or the fluorescing state of tryptophan. And so we know very well where those 1LA states are in gas phase and in aqueous phase and how they respond. So we've done a range of, let's say, transition energies and the excitation wavelengths, but the main parameters in the model are the center of the 1LA excitation and the orientation in the molecular configuration of tryptophan. So we can treat the environment in the microenvironments in many ways. We can put white Gaussian noise, let's say, or colored noise on those tryptophans to give us different microenvironments. So these as I showed you in centrioles, but I can also show you the data in microtubules and other structures. They're very robust, even to small changes in the microenvironment. So I hope that answers your question.
Yes, let's consider now just a section of the microtubule.
Sure.
So the different tryptophans, they experience, each experience a different environment, microenvironment? Or do you mean that you can basically disregard that because the delocalization effect or the super radiant, that will dominate all changes in environments, like even structural disorder, which you have showed? Is that the correct interpretation?
So the first part I would say is yes, you can treat for tubulins in solution, let's say. Let's start with the more homogeneous case. You have 8 tryptophans in a tubulin, and you're averaging in your experiments, particularly the steady state ones, over a volume that is much, much larger than the single tryptophan, a single tubulin molecule, right? So you have a relatively homogeneous environment, although the different tryptophans in the single tubulin do experience different local microenvironments. But we've been very conservative with treating the transition dipole element, which can be much, much larger, let's say the oscillator strength of the 1LA transition. So we've been very conservative in estimating in that spectrum of microenvironments what the potential strength of the oscillator is, even when excited weakly. So you're right that delocalization, I wouldn't say due to the superradiant effects. These are long-range couplings that are merged from remembering that 280 nanometer excitation involves many, many of these tubulin molecules. So if a microtubule on average in our samples is between 1 and 2 microns, so 280 nanometers compasses about a third of that, let's say. So you do see saturation at 1 to 2 microns, but on the scale of a single tubulin, the electromagnetic field is going to be pumping all of those. The electromagnetic field in a region of 8 nanometers or less is going to be more or less the same, right? They're all going to experience a similar electric field that is then going to excite them in a similar direction if they're oriented with that field. So the observation of driving them in the superradiant regime, we see that in the UV for these systems, but then we see saturation at the scale of a micron. Does that make sense? That we have in this regime where our system sizes are comparable to the excitation wavelength rather than invisible photosynthetic complexes where the system sizes are, let's say, smaller than your excitation.
Yeah, very interesting. Just a quick follow-up question. What's the typical delocalization length? Or does it scale with the size of the system that you investigated?
How do you define your delocalization length? As a single, are you defining it in a Forster sense or in some other sense?
Well, I guess that you have considered strong coupling. Let's say that you're forming excitonic states of all the coupled dipoles. So, and if you have emission from the lowest exciton states, how many dipoles or oscillators would typically be involved with that emission?
Yes, so that was shown on the previous slide. So even in a microtubule, you have on the order of 10,000 chromophores participating. Now that's what we have experimental data for that I showed you in steady state and ultrafast. But if you're interested in these larger structures, there are even hundreds of thousands to millions of chromophores, but we don't have experimental data due to the difficulty of getting, appropriate sample purity and other things, but we're working on it.
Wow, that's really fascinating. Yeah, thank you very much, Philip.
Thank you so much, Char, for your questions.
Are there any other questions for the speaker?
Sorry, may I ask a question? Kind of again with your tubulin and your centrioles, you talked about some kind of robustness, kind of as you increase the system, kind of, I didn't quite get that. Do you mean that there's a physical attraction, that there's a self-ordering mechanism in this whole process? Is there
So-
some attractive force? Kind of, sorry, I'm not very much into all your physics formulas, so I didn't really
No.
understand what that was about. But
That's
I
okay.
just, on the biological level, I'd like to understand what this actually means.
Yeah, so I didn't describe the actual self-organizing processes, but I take these, or we take these structures as given, right? So we know the structure of a microtubule well from biology, and we know the structure of a centriole well from biology,
Yes.
right? And we know the even atomistic structure of the proteins that make up these structures, right?
Yes.
So imagine
all
those
that I understand, yes.
Okay, so I was saying is then we take these structures and we look at these cooperative effects, quantum optical cooperative effects in the smaller structure and the larger structure. And what we notice, without describing how they self-organize, at least in this presentation, we then show that there is a robustness as you go to larger and larger system sizes. So contrary to this notion that quantum effects are washed out in large systems, we are finding that there is a robustness to disorder, meaning the quantum effect scales much more strongly with system size. Let me restate that. So as you get to bigger system sizes, meaning more microtubules packed together, let's say, and it's not only microtubules, but other proteins that contain this molecular chromophore, but as you pack them into the excitation wavelength of the two-level system, you then see quantum effects that are robust to the thermal environment or near thermal environment of a biological system. So,
but it doesn't mean that that it's robust on the molecular level. So it doesn't have a backward coupling kind of to the molecular structure. So it doesn't.
No, indeed it does. Indeed
them.
it does. Indeed it does have a coupling because these electronic states are coupled to vibrational modes in the proteins. So because these electronic states have a coupling, once you have an excited state, we recently showed this in publication PNAS Nexus that we published. You can find it on our website. We can show that if you excite these tryptophan chromophores, they actually vibrate down as a Jablonski diagram. You can Google it. You can then see these vibrational states in the protein that then emerge in a collective mode out of equilibrium. So these electronic states, this is a separate line of research, which I didn't show today, but those electronic states then couple to vibrational modes, which can then influence self-organization. So excellent question.
Good, thank you.
Thank you.
So it's all very, very interesting. It seems like nature tries to make itself a metal in an electronic sense.
Oh, it's interesting you called it a metal, Lawrence, because these electrons they're not flowing, let's say. These are tightly bound. You can think of them in the sense that if they started to flow, you might exhibit, and there are some proteins, I'm sure Chart knows more about this than I do, metalloproteins. There are many sort of metals that form in ions and that are co-located with biological complexes. So there are proteins that have literal metals like iron,
Right.
but there are others that actually do conduct and they produce that effect. Here, though, we're not talking about sort of that kind of electronic flow, but we are talking about optical effects that can be enhanced due to tightly bound electrons.
Right, and that's like if you look at it like the Drew model for conduction in metals.
Exactly.
Yeah,
exactly.
So that's what I mean in the sense of to behave like a copper filament, a copper wire, because I guess it's not the same electron. So if you look and see, is the electron at 0.0 and the electron at whatever length down the wire, it's not like the electron makes its way down the wire. The whole thing sort of biases to one, I guess up the field. So, well, it's all very interesting. It's all very interesting, and I'm glad you mentioned the effect of the sort of the electron photon coupling.
Yes. Yes.
But can you comment just briefly on the role of dynamics?
Mm-hmm.
I mean, I guess the coupling is a dynamic effect. But if you somehow introduce a bond that was looser, I guess, or an impurity in the system, how does that affect the superradiance?
Yes. So there are many ways to test this. So there are other chromophores in proteins in particular that do not absorb as strongly as tryptophan. Remember, tryptophan is the most strongly fluorescent and absorbing reporter in biology. So, well, compared among the protein chromophores. And so there are others that absorb at a fraction of tryptophan in the same wavelength, and they're structurally similar enough that you can mutate proteins to actually have this other chromophore in place of the tryptophan. So indeed, we're actually pursuing that with our collaborators in Europe to actually mutate proteins. They have an existing cell line that takes these microtubules, they mutate, and to see and test what functional differences, not just biological, but optical functional differences we can see even in living cells. So you asked about dynamics. So I presented very briefly this time-resolved kinetics for using femtosecond transient absorption spectroscopy. And you'll notice that the energy is going, out of the system, right? Now it's going to the electromagnetic field in tryptophan in solution. But in microtubules and proteins, you see the sharp decay, and not all of that is going to the electromagnetic field. Some of that is going into the vibrational modes of the protein. So on a very short time scale, you have this kind of burst of energy that's going into the protein that potentially could be used for self-organization processes or vibrational processes to coordinate dynamics at much longer time scales. You see, because the nuclei are, of course, much slower than the electrons.
Oh, that's great. I mean, it's decoherence and dephasing is topics that animate me.
Yes, and it's dephasing in the electronic degrees of freedom, but how does that feed coherence in the vibrational degrees of freedom, right? I mean, there's all this debate between, is it electronic coherence? Is it vibrational coherence? And I'm not getting into the debate, which Chart knows very well. But the real question is, then we know that out of equilibrium, you can drive vibrational modes into a single vibrational mode. So this is what's been known as Froelich condensation, or even Bose-Einstein condensation can be mapped, or it's a Bose-like condensation. So you map these bosons into this single state, which then coherently pumps the protein. And this has been actually observed in bovine serum albumin in 2018. Yeah.
Okay, well, we've gone over time, and I always admit that I'm the worst conference chair because I get involved in the analysis and discussion and forget about the timing part. But let's thank the speakers, all three speakers again. And we have Chart. What is the schedule like again today? Oh, that's it.
Yeah, that concludes today's talks. But maybe you could just invite everyone to tomorrow's discussion. It would be great to have many people there.
Yeah, so tomorrow we are going to be discussing sort of organizing biophysicists on the continent and globally. It's a global enterprise. And exactly what are our thoughts towards creating an African biophysical society, and what things we might do as an organization as we try to develop the organization into a global force for the things that professional societies do: member services, advocacy, prizes and recognition, and publications. It's sort of the canonical things that professional societies do. And so that's what we'll be discussing tomorrow at, I think it's 1600 GMT +2.
Yes.
And so that's 1400 GMT. And we will convene then. And then we'll send out a reminder for the session and a little bit of background material later today. So we'll see you tomorrow. Thanks again to all the speakers and to everyone who stayed around for the session all day.
Thanks so much, Lawrence. Thank you,
Okay.
Charlie. Okay.
All right, we'll see you.